Hi, this is Lance Guzman of Team 4, and this is our introduction. We're working on neonicotinoid resistance in bees.
Yay! Hi!
Yay.
Hi, my name is Vanya, and I'll be giving a brief introduction as to why we chose to modify honeybees. So this is a really critical issue in our world because honeybees are pollinators. They're one of the top pollinators in crops such as cotton, maize, different fruits and vegetables, as well as coffee, which is really important. I think we can all agree that coffee is necessary for humans, are in danger because bees are dying due to the pesticides that are being sprayed on crops. So human population is projected to grow at 80 million per annum, increasing 35% by to 7.7 billion by 2020, and about 75% before leveling off at 10 billion. So our population is constantly increasing. That means that our food production has to also increase in order to supply and provide food for everybody. But if bees die, then that would be impossible, and people would also suffer from terrible consequences, and humankind would not be able to survive. So that is why we chose to address this issue, because food is important.
Hello, my name is Bian Baledo, and I'll be explaining the rationale behind our report for genetically engineered bees. The rationale as to why it is necessary to genetically engineer neonicotinoid resistant bees is because of their vital role of not just as pollinators for plants, but survival as well. Honeybees as pollinators help disperse pollen from plant to plant when collecting nectar for their hives. Doing so helps plants to bear fruits and crops, which is as a result helps sustain the agricultural industry worldwide, which is $14 billion worth here in the US. About 1/3 of the produce we humans enjoy eating are entirely dependent on the service honeybees are doing for millions of years. Since 2006, many beekeepers were alarmed that in the US, 10 million beehives, which is worth $2 billion, were wiped out by a condition known as CCD, also known as colony collapse disorder. Some factors that contribute to CCD include parasitic varroa mites that feed on the bees, or perhaps habitat loss by metro-urban expansion or expansion of agricultural lands. Most notably, though, scientists identified a chemical found in most pesticides used by farmers to protect their crops from insect pests called neonicotinoid. It's been observed that this chemical may induce toxic and harmful neural effects to bees that hinder their survival. If this trend continues, 37% of bee populations are rapidly declining, with 9% facing extinction, according to a recent UN report in 2015. In order to play our role to save the honeybees, we decided to tackle one factor that contributes to the decline by inserting a neonicotinoid gene found in whiteflies or wood flies into the gene of a honeybee. That way we can ensure the produce found commonly in groceries will not disappear thanks to the service done by our bug friends, the honeybees.
Go.
Hi, my name is Lance Guzman, and I'll be talking about how we're going to incorporate the neonicotinoid resistant gene to the bee's genome. To start off, we would need to isolate the DNA or the gene, the cytochrome P450 gene, from the bacteria Gillomela apicola. To do this, we would need to mix the bacteria with detergent in a tube. Then we will place the test tube in hot water at 55 to 60 degrees Celsius for 10 minutes. Both the detergent and the hot bath will dissolve in the cell walls of the bacteria, which frees the DNA. After 10 minutes, we'll remove the tube from the hot bath and add about 1 centimeter of alcohol into the test tube. Because the DNA isn't soluble in alcohol, all the components of the mixture, except for the DNA, will stay in the solution, while the DNA precipitates into the alcohol layer. Then we'll let the solution sit for 2 to 3 minutes without it being disturbed. The white DNA will precipitate out into the alcohol layer. As this picture over here represents the process that we need to do to isolate the DNA. Because our viral vector genome is too long, which is 130 kb, we will use a shuttle vector, PFB-LIC-BSE, which is this, to insert our CYP450 gene. Both the CYP450 gene and our shuttle vector are digested with the use of the restriction enzyme BAMHI. This will produce sticky ends. We will then mix these two in the test tube, which will allow the sticky ends of the vector and the CYP450 DNA to bind to each other to form our recombinant molecules. The DNA ligase will then seal the backbones by creating phosphodiester bonds at the junctions. Now we will insert a genetic marker to the shuttle vector. We will be inserting the green fluorescent protein, or GFP. We will be using the same method as earlier, but with a different restriction enzyme. We will be using NCOI restriction enzyme that both the vector and GFP have cut sites for. Now that we have our desired genes incorporated into the shuttle vector, we will now use back-to-back bacillus expression system to connect our shuttle vector to our viral vector.
Hi, it's me again, Vanya. So I'll be talking about the last step, or the last part of the procedure. So after our genes have been incorporated into the shuttle vector, we are then going to take the shuttle vector and insert it into the viral vector. In order for our viral vector to be able to work in the honeybees, we need to use a promoter that is compatible with that. So we are going to add the LOC550692 promoter, which is the honeybee promoter. We insert both the promoter and the shuttle vector into the nuclear polyheterosis. And once this step is completed, we then take the viral vector and insert it in vitro. This is our viral vector, the nuclear polyheterosis. So we're going to use a syringe to add the vector into the eggs that the queen bee lays. And after they are inserted and they grow, the new offspring will take in the plasmid, and we are going to be able to recognize which of those eggs took in the plasmid because they will glow in the dark.
Hello, my name is Peter, and I will be talking about the effects of our organism modification. One result of the expression of the cytochrome P450 gene is the resistance of honeybees to neonicotinoids, of entire colonies of honeybees to neonicotinoids. An entire colony of honeybees becomes resistant to the neonicotinoid toxins by insertion of the gene into the queen bee. Once the gene that causes resistance to neonicotinoids is fully incorporated into the queen bee's genome, the queen bee can safely be exposed to neonicotinoids. The queen will lay eggs, which, too, will have the queen's entire genome incorporated genes and all. This will result in an entire generation of honeybees that can express the CYP gene, and thus survive exposure to the neonicotinoid carrying pesticides. Some of these offspring will be queen bees themselves, which will be able to produce neonicotinoid resistant colonies of their own. At the biochemical level, the effects of this organism modification is the production of enzymes that are capable of breaking down neonicotinoids into non-toxic compounds. Another effect of this organism modification is the end of what is known as colony collapse disorder, CCD. All colony collapse disorder is when all the worker bees in a colony die, leaving the queen bee, a few nurses, and all the food. All of this leads up to the final overall effect of the gene expression in this particular organism modification, the food. Since the pesticides used to protect crops from harmful pests would no longer be harmful to honeybees, food production will not decrease because bees are responsible for most of our nation's food production, approximately 80%. And without them, there would be famine. and severe economic consequences as well.
Hi, I'm Daniela Flores, and I'll be discussing the ethical and moral problems concerning modifying a bee's genome. Okay, so the ethical and moral problems when it comes to genetically modifying a bee's genome include the fact that it could result in the activation of unwanted mutations. This could put bees in even higher risk of extinction, which would create a domino effect on the world's ecosystem. Bees pollinate around 80% of the Earth's crop species. This feeds 90% of the human population. Therefore, if we were to modify a bee's genes and the result that is expected is not acquired, but rather the bee becomes mutated, this could result in a worldwide famine. Morally, there are problems in mutating a bee, as many people believe that things should be left as they are. Questions would be raised, such as who are we to re-engineer a creature's DNA when they are already perfectly functioning organisms, as well as being accused of having a God complex. But what people need to understand is that it was us humans who created the problems bees are facing at this moment, and therefore we should be the ones to fix it. And to fix it would be by making bees resistant to pesticides containing neonicotinoids.